Okay, hello everybody and welcome. So today I have, an honor and a privilege just to introduce a luminary in our field of cognitive neuroscience. So I'm honored today to have Professor Josef Rauschecker. So our theme this year is focused on the origins of language, numeracy, and thought. And we have the PhD in educational neuroscience, and we have our annual lecture series, our distinguished lecture series. And this year, I mean, we through the years have invited top leaders in the field of cognitive neuroscience. So Dr. Rauschecker is a professor at Georgetown University, and he also has a position in Germany as well at the Hans Fischer Senior Fellow at the Institute of Advanced Study at the Technical University of Munich. So Professor Rauschecker studied in Munich and Cambridge and earned his PhD from the Max Planck Institute for Psychology or Psychiatry in Munich. He moved to the United States as senior investigator at the National Institute of Mental Health. Dr. Rauschecker has also held visiting appointments at Harvard Medical School, at the Rockefeller University, the Salk Institute, and the Helsinki University of Technology. At Georgetown University Medical Center, he was involved in establishing the first fMRI research facility on human subjects. With 35 years of experience in systems and cognitive neuroscience, more than 25 years of experience in animal electrophysiology, and 15 years of experience with functional magnetic resonance imaging, he now directs the Laboratory of Integrative Neuroscience and Cognition and the program in cognitive neuroscience and cognition. He's partnered with various different institutes in various different countries. So his work has focused on the plasticity of the central nervous system. In specific, he focuses on the auditory perception or deprivation in language. He also studies the structures of the auditory cortex in non-human primates, as well as the neural basis of language, music, and other higher auditory processes, and the impact of sensory deprivation, for example, on deaf and blind individuals. His research has had continual support from NIH, the National, and also the NSF, the National Science Foundation. And Dr. Rauschecker has received various honors. In 2002, in Germany, he received the Alexander von Humboldt Prize of the Federal Republic of Germany. And there's a clear connection really between our theme, the origins of language, numeracy, and thought, and his work. So please join me in welcoming Professor Rauschecker. We're really, really pleased to have you here today, sir. So it's an honor. Thank you. And you have the floor.
Thank you so much, Clifton, for this very kind introduction. And I'm very pleased to be here, see some old friends, and at this world-famous place. Gallaudet is really an amazing place, and I've been reminded again today, getting the lab tours and talking to the students, how amazing it is. It's really unique, and I congratulate you I'm so proud of the work that you are doing here. So I hope I can scratch the surface of some of the questions that you all are working on. I think we're all interested in language, one way or another. And we're interested in how language came from, as a cognitive ability, as sort of the epitome of our human existence in a way. And animals don't have language. We might be talking about the bee language that Carl von Frisch got the Nobel Prize for, various communication systems that exist in a number of animal species, where they can indicate the location of a food source or the location of a friend, and they can socialize with each other by using communication signs and so on. But none of them has really reached the level of language. Yet, it's not going to be a solution if I ask you where language comes from, and you say as an answer, well, it came out of the blue, came out of nowhere. That's not a good answer. We, as scientists, we're not satisfied with that. So it's sort of like saying, where do babies come from? And then answer, well, the stork brings them. That's sort of an old wives' tale that people in Europe in particular used to tell their children, because they were not supposed to know where they really came from. But as scientists, we're not satisfied with these kinds of answers. We want to know about the mechanisms. So where does language come from? Are there any precursor mechanisms in the animal kingdom? Big scientists like Francis Crick, who got his Nobel Prize for discovering the basis of life, always stayed out of science. He dabbled in neuroscience when he kind of was getting older, and he made a number of ingenious contributions. He talked about sleep and dreams and about plasticity in general and all kinds of interesting questions, but he always stayed away from language because he said, well, language is, the trouble with language is there are no animal models. So, what he was trying to say, if there's no way of verifying the biological basis of something or getting closer to the mechanisms, then it's just very difficult to study. But then again, we would ask, what would Darwin say, And is there a noise coming? Oh, the technician. Like this? Is it better? Maybe you can just turn up the volume. So what would Darwin say? Is this better? I don't want to shout at you. So Darwin had his own ideas. He was attacked when he came up with the theory of evolution on his first book, Origin of Species. He was attacked by many people, of course, for some religious reasons, but also for scientific reasons to some extent. People didn't think that language would ever be able to be explained by evolution. And he stayed quiet for a long time. And it wasn't until he published his second book, Descent of Man and Selection in Relation to Sex, when he finally said his opinion about where language came from. And he had a very moderate point of view, and actually quite a modern point of view, in that he said, well, language obviously has many components. It's not one monolithic thing. It must have come from various gradual developments of the brain that did different tasks. So he was talking about hearing, he was talking about speech production, and various other components that, of course, at that time not so much was known about the brain yet, but in modern day language now, modern day research on the brain, we can assign some of these components that he was talking about to different brain systems. And now I think the question of where language came from becomes a little easier to tackle. It's still a very difficult question, but at least I think we can use animal models like non-human primates, who are our closest relatives, to come up with partial answers to some of these precursor mechanisms that we're looking for. Of course, birds have always been studied for, bird song is another good animal model of language, maybe. But in evolutionary terms, the bird's brain is often very difficult to relate in terms of homologies to the human brain. So the monkey brain is much more similar to the human brain, and there are communication systems that monkeys use, and the question has been posed for a long time, including by Darwin eventually, whether these communication systems have anything to do with language. And then, I want to start with anatomy. There are brain systems that we've identified in the monkey brain, and we do parallel studies in humans where we look for the same brain systems. And it turns out that the anatomy of the auditory system, which is the most common system that we use for communication, then has a lot of similarities. And the same is true for the visual system too. If you use a visual language, you also, monkeys use, non-human primates use very similar brain systems as humans do. And this is something to build on. So one of the discoveries that we've made over the last 20 years or so is that there is a dual stream pathway system both in the auditory system where we worked as well as in the visual system where it had been found much earlier. And those two streams or pathways have different functions. So the so-called ventral stream, which in the monkey projects, in the auditory system projects down from the primary auditory cortex into the anterior superior temporal cortex and then up into the prefrontal cortex, seems to be involved with the coding of auditory objects, meaningful sounds that can be assigned a particular role or meaning. Whereas the pathway that projects up to the parietal cortex and then via the premotor regions to the prefrontal cortex, a different part of the prefrontal cortex, is more involved with spatial analysis and, as it turns out, also with sequencing, order of stimuli. And that seems to be the same in both systems, in the visual and the auditory system. So, and it seems to be the same in the monkey and in the human. So with modern imaging techniques, for example, functional MRI and so on, we can now do experiments in humans that mimic those that have been done in the monkey and then verify via anatomical methods that these are the same systems that are doing the same function. This is a kind of the approach that we're taking, studying two primate species, one that has language, the human, and one that has a communication system, the non-human primate, the rhesus monkey. Other people are studying the marmoset, which is also a very communicative monkey species, and then relate the two species to each other. So I already alluded, my talk is going to be mostly about auditory languages, but I know here at Gallaudet you're mostly or very interested, specifically interested in, well known for sign language communication, and can we sort of make the same arguments for visual languages that we make for auditory languages? And I think the answer is yes. As I already alluded to, there are visual pathways that parallel those in the auditory pathways, and we actually had a nice discussion in one of the student groups earlier today where I was asked, how is it? Is the ventral and the dorsal pathway in the visual system the substrate maybe for sign languages? And although, we got into an interesting discussion because, of course, in sign language, there's a lot of motion involved, and the motion pathway is not strictly speaking a part of the ventral pathway. So one has to maybe assume that there's a specific role for motion of movement in sign languages. I think when the answer overall is the same. So we have a ventral visual pathway that can code the shape of hand gestures, for example, and we have a dorsal pathway that can code the location of the gestures with regard to the body. And we can go, hopefully in the discussion we can go into that in more detail and go through some of the phonological processing in sign language on the basis of these anatomical details that I will provide. But let's go back to the auditory system. And when we set out to study the monkey auditory cortex about 20 years ago, very little was known actually about the auditory cortex in non-human primates, except that there was a primary auditory cortex somewhere here in the lateral sulcus, deep in the sulcus, buried in a deep fissure, which in humans is called the Sylvian fissure. So there are some really spectacular parallels in terms of how the brain is organized in the temporal lobe, and we started to set out to map these regions around primary auditory cortex with first simple and then more and more complex sounds. And the question was really to get at the processing of communication sounds in monkeys. And I want to maybe skip ahead a little bit. No, well, maybe not. I'll show you examples of these communication sounds in a minute. And suffice it to say for now that what we found in these initial studies were 3 areas on the exposed surface of the superior temporal cortex. And these are referred to as the belt areas because they are surrounding the primary areas, the core areas, like a belt. And what you can find with some very simple stimuli, like not so much tones, but tones that have a narrow bandwidth, so we call them bandpass noise bursts, because they have a pitch, but they are more broadly organized in frequency than a pure tone that has only one frequency. And if you use these tones, these complex tones, then you find that each neuron that you encounter has a specific best frequency, and these frequencies are organized in a kind of a linear fashion where they increase in one direction from here, say you start here, then this is the increase that you see, or in the other direction you also see an increase, and then at some point the frequency will go down again. So there's a clear map of these best frequencies on the surface and in the depth of the auditory cortex. And this defines different areas. And then the next step was that we were looking for specialization for these areas. Do they prefer monkey calls over other sounds and song? So this is the kind of approach that we as a simple-minded physiologist took. So here are these bandpass noise bursts. You can see the same center frequency for all the bursts, but the bandwidth is different, and the neurons in the lateral belt prefer these slightly more complex sounds over the pure tones. So that was a big step, because now we were able to drive all these neurons. This didn't happen in the studies before us in the sort of 70s and 80s. People had tried to find and characterize neurons in higher areas of auditory cortex, but they couldn't because they only used pure tones, and those pure tones wouldn't drive the neurons. So it's like in the visual cortex, what happened in the 1960s and 70s through Hubel and Wiesel's work, when they made a discovery that neurons there respond to edges and oriented bars of light. This was a sort of a step ahead, a step forward, because we were able to drive neurons and characterize them. And the other thing, like those Hubel and Wiesel simple cells and complex cells in the visual cortex, what we found in the auditory cortex is that cells are tuned to the direction and to the speed of these frequency changes. So I might try and play a sound for you here, if I can. This is a bit loud right now. This is an upward FM sweep, and this is a downward sweep here. And you see the response of the neuron plotted here. Every dot means an action potential that the neuron fires. This is the direction of the sound in the frequency over time, and you see that there are different speeds that one can use, and only one particular speed in the upward direction triggers the neuron and makes it fire like crazy. In the opposite direction, there's almost no response. This is just spontaneous firing. So the neuron clearly is selective for direction and for speed of this frequency change. And this is essential. These are the kinds of neurons you need in order to decode a speech sound, for example. If you look at speech sounds, they have lots of frequency changes like that. There are some speech sound examples, 1, 2, 3, There's a spectrogram. If you're not familiar with a spectrogram, it's frequency over time, and the amplitude is coded by color. So you see these frequency changes all the time in natural sounds, especially speech sounds. And these are the communication calls that the monkeys are using to kind of talk to each other. And they look kind of similar. They also have frequency sweeps. They have harmonic portions in there, have noisy portions. Mark Hauser did a phenomenal job characterizing all these communication sounds in the rhesus monkey and wrote a whole book about it. And we were fortunate enough to get his library of calls when we started these studies. And I'll play you a few examples here as well. So this is a, oops, now I have to turn it up again. This is a coo call, or actually an arch stream. They're very similar. This is the coo call here. The most common communication call that a rhesus monkey makes. A very friendly call, a social contact call. When monkeys first see each other in the morning, this is what they say when they say hello, and kind of. And then you have these other calls, the grunts and the screams, that are not so friendly, of course. And you can immediately, intuitively hear that. You know, this is a threatening call. Stay away from me. So these are calls that are acoustically complex, and they have semantic meaning, obviously, even though it's very limited. You know, compared to language, this is, you could say, this is nothing. And the question is really, anatomically, and in terms of evolution, does our speech in any way originate from these kinds of systems? And neurophysiologically, let's stay at that sort of basic level for a moment. Neurophysiologically, this is the question here. So you have one neuron that you stimulate with different types of calls, and you get different responses. Obviously, the neuron likes this call much better than this call. And you see that through the firing rate. This is the post-stimulus time histogram, where you just add up these spikes over time, and you get this very nice peak, a response, as we say, to this call. So there are neurons that are specialized for particular types of calls, but we'd like to know why that is. How does the neuron or the neural network do it to make a neuron specific for this type of call and not for that type of call? This is sort of the basic question that we're asking with these kinds of studies. And long story short, we found that it's a summation mechanism. People in the neuroethology field, like Guasuga, who studied echolocating bats for many years, termed this combination sensitivity. So you have combinatorial networks that add up inputs from lower order neurons or from simpler stimuli, so the neuron becomes responsive and selective for more complex sounds in this case. So you have these bandpass noise neurons, as I showed you, which are tuned for specific center frequencies and bandwidth, and you put them together with an AND conjunction network, and you get a neuron that is responsive, selective for a bark call. This is how simple we think it is. But it's you know, you first have to wire it up that way, and the combination sensitivity or this spectral summation is a trick that is ubiquitous in all of central nervous system, especially in the auditory system, but also in other sensory systems. And you can do the same thing in the time domain. If you want to create a neuron that is selective for this kind of sequence of noise bursts, which then is by the ethologist is called a growl, one of those threatening calls, then you just have to add up those kind of simple short growls or syllables, you might call them, in the right order and with the right time delays, so you get a neuron that is selective to the call. So there are fairly simple, straightforward principles how the auditory cortex builds these selectivities for communication calls. And this is a sort of one small step towards a system that we might then later on exploit for more complex communication, that something that Darwin would have liked, I think, as a component, as one component of his multi-component system that he was postulating. Now, as I said, we go into these areas and then study many, many neurons in one area and in another area and look at what these neurons like. And it turns out that communication sounds or monkey calls are especially prevalent in an area called AL, the anterolateral belt field of this belt that surrounds the primary auditory cortex. There are three, as if you went over this a bit quickly, but if you remember the initial slide with the mapping, there were three areas, AL, ML, and CL. And so the AL is the most anterior one of these three areas. And that's the one where we find the most cells that are selected for communication calls, shown by the yellow triangles here. Back here in the caudolateral area, there's almost none of that. These cells, by contrast, are highly selective for the location of a sound. So if we play in a free field experiment, play these complex calls from different locations, they will only fire, the neurons will only fire if they come from a particular location, but not from another one. And we've seen some that are really sharply tuned for, if you just move the speaker by a few inches, then the neuron will stop recording, stop responding. So we have two very different preferences in these belt areas, and they are sort of the basis for this segregated stream system that I showed you at the very beginning with my first slide. The dual streams start there. The ventral stream starts in area AL in the monkey, and the dorsal stream starts in CL. That's very clear. Now, where do these areas project? We've done more anatomy in collaboration with Pat Goldman-Rakich, who unfortunately passed away several years ago as a result of a traffic accident. But at that time, she was still well and alive and had a great postdoc, Les Romansky, who worked with her on the anatomy of prefrontal cortex. And I asked them, I said, could we do an experiment where we inject tracers into those two areas that we just had identified, AL and CL, and see where these cells project. So neuroanatomical tracers are kind of fairly straightforward to handle, but it's a lot of work to process the tissue. So we injected these areas in my lab at Georgetown, and then Liz took the tissue with her and counted all the cells, and what it turned out is that the CL injection with one tracer led to label in this region of prefrontal cortex, and the injection into AL led to label in this region. And this happens to be dorsolateral prefrontal cortex, and this is ventrolateral prefrontal cortex. And the same two regions had been identified in the visual system as the neural substrate for visual object memory and visual spatial memory. So again, there was this match between the two systems projecting from these early sensory areas into high-level prefrontal areas that are doing the same thing. This is one system and one pathway. This is the spatial pathway, and this is the object pathway. And they can be shown in both the auditory and the visual system. So this was, I think, our first really big accomplishment. This is where we started to begin to understand these systems. And then we put things together, and there was more mapping to be done in the various regions. This was then a PNAS paper that we had in the year 2000 where we put this all together. And different projections from the different parts of the thalamus, the medial geniculate, dorsal and ventral part. And in the meantime, we know a little more about the organization of the ventral stream. We're still working on all of this. How do these communication calls then get processed further? How do we achieve categorization, pitch invariance, all these things, all the same questions that we're asking about human speech and language as well, can be asked in a monkey at the neural level now. And the same thing happened with the dorsal stream. And of course, that story took us a little somewhere else when we started to do human studies. But first, one other detail, there's lots of techniques available now. One can do functional MRI in monkeys, of course. One can train them to lie in a horizontal MRI scanner and then listen to the sounds, and you get beautiful pictures, and I'll show you in a minute. But there are also microstimulation techniques which we cannot use in humans. So there are a number of techniques that can be used in animal studies that are not available in humans, and they give you a lot of information about connectivity as well in a very concrete sense. So this was a study that we did in collaboration with Nikos Logotidis and Chris Petkov in Tübingen at the Max Planck. And we did microstimulation of the ventral stream, first stimulating in the anterior part of the superior temporal gyrus, which we knew was selective for monkey calls, and trying to look at the activation in prefrontal cortex, basically replicating the anatomical results with physiological techniques. And we found some activity, but we were kind of disappointed that it wasn't more. So we went one step further and stimulated in the STS, which was sort of a little more lateral, and we thought maybe closer to prefrontal cortex. And sure enough, we get much more activation when we do the stimulation from there. So there are distinct advantages of using animal models to characterize the functional organization of these pathways, which we think are basic and are important for the evolution of speech and language. On the other side, in the dorsal stream, we also used functional MRI. So this is an example of how you can use functional MRI techniques that we can also use in humans, in the monkey. And this is kind of a bridge technique, as it has now turned out, that gives us results in both species that we can then relate to each other. So in this case, we studied the organization of space processing, and we found that indeed, again, as we had found at the single neuron level, posterior regions of the auditory cortex are more selective, are more prone to processing the location of a sound than more anterior regions, although we didn't find a more detailed space map as we had hoped. Now, back to the vocalization pathway. This was one of the earliest functional MRI studies that we did at Georgetown with our own scanner. These are, don't tell anybody, but these are the same scanners that are also used for humans. And we just have to use a different coil. And it's the same technique, basically, that we use in humans, same field strength. And we get beautiful results here. This is the ventral stream, clearly, as you can see, and almost like a pearl, like a chain of pearls, these areas are lined up now that are responding to monkey calls much better than, for example, to environmental sounds that we used as a control here. So environmental sounds are good stimuli as well. A food cart that you roll by the monkey room, of course, will get noticed by the animals, but it's processed in a different region than where the monkey calls are processed. So we have a pathway that is obviously of relevance for communication sound processing, and then develops into maybe something like a speech pathway eventually. So, and we were looking for hemispheric specialization, which we didn't find. In the monkey, there is bilateral activation, and maybe this is one of the distinctions that made language more and more usable. We don't know why it has to be left lateralized. There are various theories about that, but monkeys don't have that lateralization yet. Now, as I said, now we can go to the human and then start over and see what the auditory cortex is like there. One of the early studies that we started with this technique was, I'm losing this. Why did that happen? Yeah, this is it here. Okay, so what you can see here is that the human auditory cortex is organized in the same way as the monkey cortex, with a core and a surrounding belt, and then another layer, the parabelt, which is sort of the tertiary layer of auditory processing. And when we used simple speech stimuli like vowels in comparison to bandpass noises and pure tone bursts, we find this very clear segregation of activation, whereas the early areas essentially respond to all three categories of sounds. Vowels are only processed in the outer shell, in the parabelt regions, which are pretty far along the ventral stream. And so we can also now map functional topography of auditory cortex in humans. And Amber Lever, who was a graduate student who did a number of very ingenious studies, she was able to map the complete auditory cortex in humans with the different regions that we had discovered in the monkey, with the belt areas and the parabelt areas, and related to the known anatomy that was done 20 years, almost 20 years earlier in a lab in Germany. Morosan and her collaborators had found cytoarchitectonic differences which kind of are much more precise in a way than the Brodmann atlases that only distinguish between, let's say, A1 and area 41 and area 22, if you know this nomenclature. So there are much more distinct differences between the various regions around Heschel's gyrus, and they correspond to functional regions that we can identify with the functional MRI technique. And the two are now matchable quite in great detail. Now, other people have also picked up on our proposal of dual pathways and of the different functions, and Robert Satori, for example, did extensive studies on auditory object perception and sound localization, etc., and he found that this anterior region of the superior temporal cortex is especially important for the processing of complex sounds that can be identified as objects. People were actually trained in recognizing and distinguishing different sound objects, and then they did the imaging, and sure enough, it's the ventral stream again that lights up. So the ventral stream is not just for speech and language and communication sounds. It is good for all types of complex sounds that can be related to particular meaning or to semantics. So it's an object pathway, as we say in general, and that matches very well what is known in the visual system about the function of the ventral stream there. By contrast, the dorsal stream, again, is important for space and motion in these human studies. A group in Cambridge has done these studies also more than 10 years ago now, where they replicated basically the finding in the monkey where stationary sounds that are presented in different locations generate activation in this posterior part of the superior temporal gyrus and up into inferior parietal cortex. And it's actually, and this is maybe important for the purpose of understanding how sign languages are encoded, the activity is strengthened, is emphasized even more when the sound is moving in space. So this is where I would predict moving gestures are especially prevalent as preferred stimuli. So if we put all this together, we're looking for, is there a region that is special for speech? Is there a region that is special for music, for example? And again, Amber Lever, in one of her studies, when she was a graduate student in a lab, found that, yes, here's primary auditory cortex, and there's belt cortex out here. It's a horizontal view. And there are regions in the lateral superior temporal gyrus, especially in the anterior, on the anterior side, exclusively on the anterior side, as one would have predicted, where speech sounds are especially good stimuli. So this again matches the monkey. There are neurons or neuronal patches that are tuned to speech more than anything else. And maybe there is a subsystem in there in the ventral stream that is specific for speech sounds, but it is embedded within other object categories that are also characterized by complex sounds. Musical instrument sounds are represented somewhere on the right side, and here you do see the lateralization. Music is on the right and more anterior. That's what other people have also found with PET studies, like Robert Satori, where a study where he finds exclusively right-sided activation by music sounds. And it may also have to do with the temporal and the frequency, the spectral structure of music compared to speech and language, which has sort of faster components in frequency processing. A study that came out recently from MIT, Nancy Canwisher, who discovered the fusiform face area and many other things, and one of the leaders in cognitive neuroscience, she now gets interested in in auditory cognitive neuroscience and is interested in speech and music. So with her collaborators, a student in the lab, and Josh McDermott, who's been around the block also for a while, has collaborated with Mark Hauser in the past on monkey vocalizations. They've set out, did a new study on specialization of auditory areas in the human cortex, and they replicated our two areas that are specialized for speech and music. So speech is the green area here on the lateral side, lateral to the primary auditory cortex, and then music more anterior and a little more medial. So this is shaping up as, we're trying to do the same thing that people in the visual system have done for a long time. Regions of the sensory cortex are specialized for certain functions. And it's just, a matter of finding out what they're specialized for. We don't have a good handle unless we have, the monkey studies, the anatomy that we have gained from there, and maybe other, lesion studies as they were done beginning with the 19th century, all the gunshot wounds, how it led us to, in World War I and in the Civil War here in the United States. This was actually the earliest body of knowledge that we have about functional specialization in the brain of humans. And now we can relate this to the functional imaging data and to the anatomical data that we have in non-human primates, and then we're beginning to find a much better way of characterizing all these regions. Now, at some point, we got really bold and we started to use speech sounds in our characterization of the dorsal and the ventral stream. Ian DeWitt, who is now at NIH, was a graduate student with me at Georgetown, and he started out to compare activation between really simple speech sounds, phonemes or syllables, you could say, phonemes, let's say phonemes, words, and then short phrases, and he also wanted to get into sentences ultimately. And he decided to do this on the basis of a meta-analysis. So he took dozens of other studies that had been published on speech processing in the human brain, and what he found was quite astonishing and quite interesting. So the simplest of these speech sounds are processed closest to primary auditory cortex. This would be what in the monkey we call anterolateral belt. So the early stages of the auditory ventral stream. And then as you move anterior and you get more complex sounds that are combinations of more syllables and more complex sounds, it could be even monosyllabic words, but they are still spectrally and temporally more complex than a simple phoneme. You move anterior. So this is clearly more anterior than the phoneme representation. Then as you go even more anterior, you find selectivity for short phrases. These are only really sort of 2 or 3 word brief sentences because, longer sentences are coded somewhere else because they need other mechanisms for activation than these short stimuli do. But this was an interesting finding which has found a lot of attention, because what it also told us is that the representation of words is not in what used to be called Wernicke's area, but it's much more anterior. And so, so what is Wernicke's area actually doing? It's not the auditory word form area that some people assumed. It's not there for the decoding of speech sounds. This is done more anteriorly in this new area that we found. I think the answer comes when we look actually at results like those from Laura and Petito's work in sign language. She did an ingenious study at McGill in collaboration with Auditory and others, and published a paper about this, sponsored in PNS by none other than Noam Chomsky himself. And what it showed is that in profoundly deaf people that process sign language, the same region that people used to refer to as Wernicke's area and were thought of as the region for decoding speech sounds at the word level in spoken languages is also activated by sign languages. So this, I think, was a real eye-opener, was a very important finding that not only did it show that posterior superior temporal cortex was probably multisensory, which it is. We've done some studies on the McGurk effect recently in our lab, and it clearly is activated by lip reading. So it's an integration area between the visual and the auditory modality. But even more so in deaf signers, they use the same region for the decoding of sign speech, and they don't. I don't think it's a decoding at the sort of sensory level. It is more important to see this in the context of now the dorsal stream. So the posterior superior temporal cortex is the beginning of the dorsal stream, as I showed you in the anatomical data from the monkey. And the same is true in humans. So the one could also ask, going back to Darwin's question of what was the origin of language, but some people think, well, maybe sign language was there first. It was probably something that people naturally had, and they used gestures to communicate, and then spoken language kind of developed, evolved on top of that. I would argue that the two forms of languages, visual and auditory languages, evolved together. These were actually part of the same mechanisms. What you need and what the dorsal stream is really doing is the sensory motor coupling of the sensory and the action information, motor information, or articulatory information. And STG, posterior STG, is the beginning of that pathway of this process, which goes up through parietal cortex and into dorsolateral prefrontal cortex. And here's another sort of line of research that I would like to tell you about that kind of bolsters this view. It starts out with an idea I had many years ago. When I'm sure you've observed this yourselves. If you know a music piece really well, it could be whatever genre of music, whatever you prefer. You, in the old days, still had your old LPs at home or your CDs, and now, of course, we all have our iPhones with the music on it. But in the days of CDs, let's say, you played a certain track, many times, and then it was followed by another track. So when you now go back to that same song and you play that familiar track, you will automatically, at the end of that track, you will hear the next track. Yeah, Tom is shaking his head. He's sort of nodding, confirming this. And everybody who I've talked to has made that observation. You turn on your car radio and if you play, oh, there's this famous Beatles song, and then at the end of the song you start singing the next one. So where is that stored in your brain? What kind of memory is that? And it has to do with familiarity, obviously. And at some point, we published something about this, and a reporter picked it up, and I told him the story that this started with my old Beatles LPs. So he gave that title to the piece that he made, that he wrote and recorded. And what it is, I wrote another paper a little later, because I still couldn't figure out how this is done by the brain. How can you have a prospective memory where your brain predicts what the next song is that you're going to hear? It's really quite interesting. And so is there a tape recorder in your head? The principle of a tape recorder is that you record something on a medium, electromagnetic tape, and something that changes over time. So the time change gets translated into a location change by virtue of the moving tape or the moving CD. It's the same thing. And then when you play it back, you reproduce that piece by turning the table or the tape in the same direction and pulling it through the reading head. So it's a mechanism where you translate time into space. But the brain doesn't have any moving parts, so how does it do that? And we found the answer by doing this simple experiment in the lab where we used Georgetown undergrads and asked them, bring your favorite CD and we'll digitize them, and then you go in the scanner and then we play these songs to you, and then at the end of each song you will just hear nothing at first. And so we imaged them while they were listening and then also while they were waiting for the next song to come. And during the silence, they showed activation in their brains, but where was the activation? It was in the dorsal stream, it was in the premotor cortex and in part of the prefrontal cortex. So clearly up in a sensory motor pathway, and we think it's not only important for coding musical sequences or sound sequences, it may be important for coding sequences, period. And this is now the new model for the auditory dorsal stream, which went back to the surprising Beatles observation. That's why I think John Hamilton's title is quite appropriate. I should write a letter to Paul McCartney thanking him for this idea. So here's the dorsal stream again, which we've characterized in the past as a spatial stream and maybe movement stream, but it's more than that. I think it is linking up the sensory information, in this case the auditory information, with prefrontal cortex via premotor cortex. And that's the whole answer why this region gets activated by speech sounds, in both the auditory and in the visual domain, and it links up to the ventral stream information, the object information, which is also inherent in both types of languages, auditory and visual languages, at a place that we used to call Broca's area. So Broca's area is nothing else than the place where high-level sensory information gets translated into articulatory information, and we now refer to this as a forward model where this is incredibly important for language acquisition, where we think the young babies have auditory information long before they have articulatory information. If you ask your 1-year-old or less than 1-year-old, where is your nose, they will be able to point to the nose. So they understand that the word nose as a term, but they will not be able to say the word nose. So there is clearly the sensory information comes first, and then it is used to build up that articulatory representation that is contained in the dorsal stream, and you have to close the loop in order to be able to process language. So here's a nice example of studying how we can now study the articulators in the speech system, in the language system. I hope I'll be able to play this. We're going to shorten this just a little bit. As much as we can enjoy this, if we hear this, I wanted it to just demonstrate to you these are the new techniques with real-time MRI, which are becoming more and more available. And they, I wanted to show this to demonstrate what a complicated articulatory system we have. It's not just the larynx that people used to talk about. It's the whole supralaryngeal system with the tongue, with the lips, with the mandibular, the velum, and so on. And if we, there's also a real-time MRI of speech, which I could show you, but I'm going to skip that. We can look at that later. It's the same thing. The tongue does a ton of work in coordination, temporal coordination with the mandibular, with the lips, with the larynx, and the lungs in particular as well, because it has to pump the air through there for us to be able to articulate. So this now goes into another direction. There's a really famous book and a wonderful book to read by Pim Lefeld, former director of the Max Planck Institute for Psycholinguistics in Nijmegen. He was writing about this 10 or 20 years ago, how the process of speaking is essential for our development of spoken language. It's not just the auditory perception that is important, it's also the act of articulating speech sounds, and that's where our dorsal stream, the whole closed loop system between the sensory and the motor system gets so important. And I think this principle holds for both types of languages. It's just as relevant for sign languages to understand. They all work on that principle in a very universal way. So the larynx is relatively simple. There has has a couple of muscles here, and it's complicated enough, but this really determines the pitch of our voice, the vocal cords, and so on. The respiratory component is important, as I said as well. It has to deliver the air that then forms our voice and our voice speech. But what's really complicated is the supralaryngeal component. This is the apparatus that really produces speech with the tongue having about 50 muscles that are innervated by various nerves. And to build such a system is just crazy, I think. How could this evolve in nature as a system for us to communicate? Compare that with the monkey system. This is so far superior. This is no comparison to what the monkeys can do. But to understand how this could have evolved, and you can make the same arguments for sign language, where you also have to have a system that does complicated motor and sensory motor coordination in time and space that is just stunning, in my opinion. So I think the answer to Darwin's question is, again, there's an increase in the number of moving elements on all sides. You have an auditory cortex that gets more and more refined and sophisticated. You add on more and more specialized areas from the monkey to the human. So you're able to actually decode those sounds that you're producing with a more and more complicated articulatory apparatus. There's a combinatorial explosion in the number of vocal sounds that we can produce as we make our vocal apparatus more and more complicated. And of course, it's the innovation also that comes down from the brain, from the premotor system and the motor system through the brainstem and so on, that has to drive this. But the elements that are moving in the articulatory apparatus also have to be there in the first place for it to be controlled. So the argument that some people have made now, well, some people have been saying this for a while. Phil Lieberman, for example, was someone who always advocated the importance of the articulatory apparatus as a driving force for evolution of speech and language. But then people said, well, it can't be that. It must be the brain. But of course, the brain does this in conjunction with the periphery. And so I think it's a co-evolution of both of these components. If you look at the number of words in English, it's 170,000. People, probably it's more than 200,000 if you count some of the other words. That's sort of the effect of this combinatorial explosion of the sounds and of the articulations that we can make. And the same thing holds to a certain extent also for sign language, of course. Now look at the two species again. Here's a 2-month-old human baby who is beginning to coo. And here's a young infant monkey that tries to do the same thing. And at that point, they're still fairly similar. They're kind of, the human baby isn't much better than the monkey is. But then it begins to change. At some point, the human babies are beginning to babble. This is babbling now. And it's still pretty simple. It's only one vowel and essentially one consonant. But it's the movement of the mandibula that is so important. They begin to make coordinated movements where they activate their lungs and the vocal cord, the larynx, and so on. So there are 3 moving parts already that allow us to make kind of human speech sounds, whereas the monkey stagnates. They never get that far even. This is an adult monkey making one of those monkey calls that I showed you earlier. And they just never get beyond that stage. So there's the difference between the articulatory explosion that we've faced and where monkeys stayed behind. It's good for their purposes. They can do communication with this, but they cannot generate 200,000 words with it. And so, we figured there must be another way to study the precursors of language in monkeys, other than making them vocalize, because they're not going to get very far. They can vocalize as much as they want. They will never get anywhere close to humans are, and so maybe we have to bypass the laryngeal system and make them produce sounds in a different way. So monkeys are good at pressing levers. They're good at motor sequence learning. They can do all kinds of motor sequences. So we figured if we tie up these levers to sounds, program the computer to actually produce sounds, then we can produce another sensory motor communication system that we can then study a little better, that we can expand in complexity. So here's one of our monkeys pressing these levers, and each lever makes a tone, and then he is trained to do this in a certain sequence, and then he learns a melody that he produces. Okay? And then we do this with another monkey, and I can maybe skip this one because this monkey was not so good at this, and this is a really good monkey that we have here, who was very articulate after a short time, although she was a little squeamish in other ways. Looking for the reward. They are dependent on the rewards. This is another difference, of course. Babies do this without looking for reward necessarily. So, but she ultimately learned to play with two hands and was really good at that. Actually, there's an example here. Doesn't always work. So where do we get with this? We do put the monkey into the scanner and do functional MRI, and then play those melodies to them that they've learned to play on this complicated apparatus. And what we find is we get auditory activation, as we should, and then we, in the auditory cortex, and then we start to compare, we get activation also in some part of the motor system, basal ganglia get activated because it's a sensory motor task. And then we start to compare sequences that they've learned to play, self-produced melodies that they've learned to play by themselves, and melodies that they only heard passively by listening to another monkey play. So that's the fundamental difference. Is the self-produced sequences are the ones that engage the auditory dorsal stream, where they have to activate the motor system as a result of sensory processing and vice versa, whereas the passively heard sequence is, presumably purely auditory. So what we found is that they activate premotor cortex in both cases. So this famous area, F5, is the mirror neuron area that Rizzolatti and others found in the visual system when monkeys grasp and reach. These neurons also activate when the monkey observes another monkey grasping and reaching. So it's another example of a sensory motor control system, but the self-produced and non-self-produced were still the same. And But then we found another area in premotor cortex, dorsal premotor cortex, F2, where there's a significant difference between the self-produced and the non-self-produced. This is the difference between the two regions. And so this is specifically activated when the monkey hears the melody that it has learned to produce by him or herself. So we think, again, this is the premotor system that we found in the beetle study that is involved in coding of these sound sequences and probably all kinds of sequences. And we're now setting up to studying these patches, these neurons in these patches with a single unit electrophysiology. And that hopefully will get us to the neural basis of some of these sequencing systems, sensory motor sequencing processes that may be the origin or the driving force for communication systems like spoken and signed languages. And now it gets really complicated, and I think I have to wrap up little by little because we still want to have some discussion, I hope. So, but here's something you already know. This is the ventral stream, the dorsal stream, as I've shown it to you multiple times. Broca's area right here in the middle. It's next to premotor cortex and prefrontal cortex. Inferior parietal cortex plays a very important interface function. It's probably the place where the motor error is measured and corrected, and the input system is here. You could replace that. Superior temporal cortex plays a role in both auditory and visual languages. So it doesn't matter whether you use spoken or sign language here. And then there's this big motor system kind of hanging on to this kind of control system that's up there. And that's kind of complicated. You have not just motor cortex and the basal ganglia. You have to drive the pontine nuclei, and then ultimately you get down to the phonatory motor neurons that are doing the actual hard work of articulation, or in sign languages, your hands and arms. And but don't forget the feedback. Without feedback, nothing works. So the proprioceptive feedback within the somatosensory cortex may be very important. And of course, for spoken language, the auditory feedback is important, as we know. And this is all, then fed back. And so now we're facing the daunting task to actually start analyzing the motor side of articulation if we really want to understand this. And this is how it works. You have the motor cortex up there controlling the motor nuclei. This is another server system where everything happens. But then the hope comes by using control theory to actually understand all this. So as I used before the term forward model or internal model, this is how it works. It's a control system where the controller, which is probably in the premotor cortex, normally during normal behavior drives the motor output, and that's where all this goes. But in order for this to happen, you have to have this feedback loop that goes through the parietal cortex and and the prefrontal cortex ultimately, and this is what we really need to understand in order to understand how languages work and how they ultimately originated. There is a little paper that we published a couple of years ago with Ina Bornkessel and her husband and Steve Small. The Bornkessels are in Australia now. They originally trained at the Max Planck in Germany, and think that the monkey studies are quite valuable for understanding language. Linguists are kind of split in their opinion about the value of these animal models, and I think the ones that are interested in the biology of language are keen to get more of these monkey studies to be done and interpret them in a cognitive sense and in terms of mechanistic models of how all these linguistics terms that have been developed over the years, how they can be realized in biological terms. And I think we've had a few thoughts in there that are quite relevant, especially with regard to the dorsal stream again, where we think sequences and languages consist of sequences at various levels. They consists of sequences of syllables that then form a word, and of sequences of words that form a sentence, and then sequences of sentences that form a whole conversation. And all these sequences have to be represented by internal models in the brain. And I think it's really how this works. And so, in conclusion, I think we've shown that anatomically there is a basis to say there are precursors of language in non-human primates. The ventral stream is undoubtedly very similar. It does the same thing of object recognition. The dorsal stream, we'll see. We'll have to see whether there is a fundamental difference in the monkeys compared to humans in terms of their internal models that they use. And I think the approach that we're taking with the sensory motor learning task, where they press levers instead of articulating, which they can't, is the right way to try this. And now we have to also, if we want to go back to language as a referential system, that's a whole new ball game. We have to look at the ventral stream again and see whether there's more than just the kind of primitive food call or social call reference that they undoubtedly have established, probably innately. And there's probably nothing much there. It's going to be difficult to find this. So far, there is no good evidence that this kind of referential system exists to the same extent. So there's the other component that may be missing in this multi-component system that Darwin had outlined. But we'll see. Here are the people who did some of these older studies with me. Pavel Kuzmarek has been in the lab for over 10 years now. And these three guys helped especially to establish fMRI in the monkey lab. Josie was a student from China who worked very hard. She's now in a PhD program in Belgium. Michael Ortiz was a graduate student from Puerto Rico who then went to Nikos Logothidis' lab with the help of our NSF PIA grant, and has now a postdoc position in Newcastle, England. And Denis Archikov is originally from Russia and now in Helsinki, Finland, and is currently helping us with some follow-up experiments with the analysis of these data. And Dan Cameron, who sits in the background here, is a new postdoc who came from Canada, who will hopefully help to move this project forward. And these are the fMRI guys. Amber Lever was a graduate student, very successful, did a lot of work in the lab. John Vermeer runs our imaging center. Those of you who are doing imaging at Georgetown will probably know John. Wonderful guy who makes everything happen. And Ian DeWitt and Mark Chevier were the two graduate students who I mentioned in my talk. And they are also moving on. Mark has now with, I think, Google or Facebook, has a well-paid position, I have to say. And Ian is still at NIH, looking for a job, actually, at the moment. The funding came from NIH, from NSF, and from the Humboldt Foundation. And if you want to hear more music examples, I can do that later on. Thank you very much for your attention and for having me here at this wonderful place.
That was a wonderful presentation. I really enjoyed that today. So now we do have some time for some discussion. Any questions that you guys may have or comments. If you guys have any thoughts that you do want to share, please come up to the stage. And just please note, after the discussion's over, we're all going to go over to the SLCC atrium. And we'll be there at 5:30. We'll have a reception over there, and we'll have a chance to talk and discuss some more. Okay, so does anybody have any thoughts they want to share or any questions? So I have a question. Should I come up? So I think I'll just sign and he'll just voice for me. So right now we studied David Koppel's The Mapping Problem, and I'm wondering, do you have a good understanding of the specific areas? And we have an understanding of what their specific areas are and their purposes. We don't know how they work together like as a system. So I'm curious about your thoughts about where the field might go and where we have capabilities to map, or when we might have the capabilities to map the full system, and how you might think that progresses.
Which system are you specifically talking about? Are you talking about language systems?
Like the language processing systems.
I'm sorry?
Sorry, language processing systems.
Language processing systems as a whole. Well, I think I was sort of alluding to that towards the end of my talk. There are two major challenges, I think, that we have to try and understand. One is how the sensory systems and the motor systems talk to each other, and how they organize articulation and expression in the temporal domain. And so, in a way, we need to understand these internal models, and this is true for both the spoken and sign languages. I think the principles are the same there. And we also need to understand how the ventral pathway and the dorsal pathway talk to each other, because obviously they are separated in terms of anatomy. They are segregated pathways anatomically, but they meet. We know that they meet up in inferior frontal cortex. So Broca's area, so to say, is this interface area, as I've called it, where the high-level sensory information interfaces with the high-level motor information. So I think we need to understand that process up in inferior frontal cortex. The other, maybe even more challenging question is how the referential system works. How do we attribute meaning to either a sound or a gesture? And that's sort of, one could simply say, well, the question is, where is the lexicon? Where do we store meaning? How do we remember information about an object that we then label with a particular sound or a gesture? But I think it's more than that. It's sort of the whole idea of, this is sort of crucial if one thinks about the evolution of language. How did the brain or nature or whoever designed this get the idea to use an arbitrary sound or an arbitrary gesture to an object out in the real world? This is something where I personally feel most challenged. I think the other part, the sensory motor part, is something we can ultimately understand. But so we talked about this in one of the groups today, that obviously exposure to language is essential. If somebody grows up without any exposure to language by other members of their species, they may not ever develop language. There are sort of some kind of poorly documented examples. Caspar Hauser was one in Europe in the 17th or 18th century, which was documented by a movie. Werner Herzog made a movie about it. But there are some other examples in the literature. And so I think language is defined by its function of communication with members of the same species. And if there's nobody there to communicate with, it might not ever develop. So this is a difficult argument to tackle experimentally, I think.
Thank you so much. Hello again. So that was a fantastic presentation. Thank you so much for the information, and also great work. I wonder what your thoughts are in situations like sensory deprivation and auditory deprivation. For example, people who have hearing loss, they use hearing aids, cochlear implants to communicate orally, and those devices introduce new distortions to stimulus. So how do you think those types of distortions match your models of auditory processing? Do you think it will fit the model, or do you think it needs to be modified in terms of the network itself?
When you talk about distortions, could you be more specific what you're referring to?
Sure, sure. So I'll use the example of a cochlear implant. The cochlear implant signal is not in any way proportional to spoken language. It's very forced, it's very mechanical,
Yeah.
it's very limited. Lots of speech information is missing from
Right.
what you're actually receiving from the cochlear implant signal. It's not really a natural language process, but it's still auditory. So I'm curious if that matches that same auditory model for hearing people or people using spoken languages, or is it something that's based on linguistic demand or more of a socio-functional demand?
Yeah, no, I think that's
executive function demand.
That's a really good
interpreter
question, Brad. And we touched upon this briefly over lunch, I remember. And obviously, it's the higher levels of the speech and language system. You know, let's just talk about, you know, in this case, it's the example is auditory perception, you know, through the cochlear implant. So auditory cortex would be the sort of the first station where this arrives in this whole processing system. And obviously, auditory cortex has to fill in a lot of the gaps. It can't be done by more peripheral portions of the auditory system. They are not sort of probably prone enough to filling in these gaps. And I think you're right. When you're saying, is it higher levels of language, the language system that are contributing towards filling in the gaps, I think that's probably correct, that there has to be a top-down process that then helps to fill in the gaps. On the other hand, if somebody does not ever had any experience with spoken words or language in the auditory domain, it may be hard for the language system to fill in the gaps. And so it's hard to imagine how this happens. We know it works, and we know it even works with people you know, outside of the cochlear implant domain. You can construct stimuli that are so-called sine wave speech, where you play speech through a specific filter that leaves out a lot of essential information. But it has the temporal information in it, has sort of pitch changes in it. It just lacks the high frequency detail. And amazingly enough, after playing these impoverished sounds, which sound sort of like cochlear implant signals, I think, especially if you have very few channels, then you learn to understand. And how this happens is still kind of not fully understand, but it is kind of in the realm of filling in phenomena, where even in the visual domain as well, we can kind of substitute missing parts. You have a figure that is hidden by another figure, and you still recognize what's underneath because you kind of fill in the gaps in terms of information that is hidden. You know, so it must be a top-down process in that respect. But I think it's something one should do more research on, for sure.
Yes, I agree. Thank you so much. Okay, great questions. Do we have time for one more, Dr. Allen? So I think my question's fast, so. Well, great. So we'll end with Tom, and I'm going to go ahead and start handing out evaluation forms to you all. If you could just fill them out. I hope to see everybody over at the reception after Tom's question. So I'm going to voice, I think.
My question has to do with timing. We know timing is very important. Rhythmicity, temporality, prosody, very important for a child to be able to determine what is language and what is not language when the auditory or the signing stream is coming in. My question has to do with the monkeys, because I noticed they played the sequences in a very set rhythm. I mean, it was very scored. And my question was, were they taught that rhythm, or did that emerge? And was that sort of a natural monkey rhythm?
It emerges.
What?
It emerges. The short answer is every monkey has its own rhythm.
So that wasn't something that you didn't teach them, that bum, 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 bum?
Well, you know, ideally we would have liked them to play in a certain, given rhythm, but it's very hard. You know, they start to develop their own rhythm, and then you can't untrain them. And we, you know, the big question was,
That's
when,
interesting. I mean, that's an
yeah,
interesting
it's
observation.
interesting in itself. But the big question was then, you know, when we started to do the fMRI imaging, we said, you know, should we play back, and we had to do the playback experiments, should we play back the idiosyncratic rhythms that they developed, or should we play back the ideal rhythm that we wanted them to play?
Yeah, well, that was my next question.
And-
Did you
and
speed it up and slow it down to find
We
out if there
didn't
was
yet,
what-
but this is something that is on our list of things to do. This would be an important control to see, you know, how much this holds over different time scales. But the short answer is that we played back the idiosyncratic sequences because we thought, you know, this is
That's
what
know.
they remember, this is what they know. But it would be interesting also to see, you know, compare this response that we got with the idealistic sequences and see whether they can distinguish them from each other. Is this the same for them? So in terms of categorization, you know, this is actually another problem in language. You know, how do you categorize the different words and phonemes and so on into one and the same semantic entity? You know, this is a totally unsolved problem so far. We know that's a top-down process that comes down from prefrontal cortex, but how the brain does that, it's, you know, it's sort of a fundamental problem for perception as a whole, but in language in particular is totally unsolved so far.
Fascinating. Very-
Okay, wonderful questions, everyone. And I really enjoyed your lecture today, and I really look forward to just talking with you more at the reception. Before we do go, I want to give you just one small gift. I really just thanks for your time and appreciation of you coming.
Oh, no. Thank you so much.
Thank you for coming, and a round of applause. Thank you again.